Good afternoon, students. Today we will study about the embryo, development of embryo, how embryo are formed. The egg cell of the embryo sac is the fertilized by the male nucleus to form a zygote. The zygote undergoes a brief period of rest which differ from species to species. However, in Oryza and Crepis, the zygote begins to divide a few hours after fertilization. During the resting stage, the zygote undergoes several changes such as reduction in the vacuolar size, increase in cytoplasm, and aggregation of ribosome to form polyribosomes. The various steps from zygote leading to the formation of embryo are called embryogeny. The embryogeny found in dicot and monocot will be discussed separately. In the early stage of embryogeny, similarities in both the dicot and monocot embryo shows. The plane of first division of the zygote is almost transverse, forming a small apical cell and a large basal cell. The two-cell structure is known as proembryo. On the basis of further development of proembryo, dicotyledon embryos have been classified into 5 main types by Maheshwari et al. In 1950. The terminal cell divide longitudinally. The basal cell contribute only a little toward the embryo formation in case of cruciferous type. While the second type, the basal and the terminal cell both participate in the formation of embryo in case of asteroid type. Second is the terminal cell divide transversely. The basal cell contribute a little toward the embryo formation. It divides to form the suspensor in solenoid type. Second, the basal cell does not divide, and if suspensor is formed, it develops from the terminal cell as in caryophyllid type. Third one, the basal cell as well as terminal cell participate in the formation of embryo as in case of chenopodial type. Development of embryo. Hansteen and Flemington's 1870 studied the embryo development in Capsella brusa pastoris, which represents the typical dicot and cruciferous type of embryogenesis. The zygote divide by transverse division, forming two cells, an upper or the terminal cell and a lower basal cell. The basal cell further divide transversely, forming two cells, whereas the terminal cell divide by vertical division to form two longitudinal cells. Thus, a 4-cell proembryo is formed. The terminal cell again divides by a second vertical division at right angle to the first one, resulting in the 4th cell and referred to as a quadrate stage. The transverse division now takes place in the 4th cell, giving rise to 8 cell, arranged in the 2 tiers of 4 cell each. It is known as an octant stage. In the octant stage, the upper tier of the 4 cell is known as the epibasal or the anterior octant, whereas the lower tier is referred to as hypobasal or posterior octant. The 8 cell of the octant stage now divide by a periclinal division, resulting in a 16 cell stage, arranged as 8 outer cell and 8 inner cell. The 8 outer cell form the dermatogen, which further divide anticlinally to form the epidermis. The inner 8 cell divide by vertical and transverse division to differentiate an external layer of the cerebellum lying below the dermatogen and a central region of the pleurome. The cerebellum develops to form cortex, whereas the pleurome forms the stem. The development takes place in the 2 cell of the basal cell divide by 4 to 6 transverse division to form long 6 to 10 cell suspensor. By the time the embryo is in 16 cell and assume a globular shape, the suspensor is fully formed and help to push the embryo deep into the nourishing tissue of the endosperm. The uppermost cell of the suspensor facing the micropyle enlarged to form a vesicular cell, the hostorium. The hostorium is normally unbranched but may branch in certain cases like in sedum, air. The lowermost cell of the suspensor lying adjacent to the proper embryo is known as the hypophysis, which partly contribute to embryo structure. The hypophysis now divide transversely forming two cells, which further divide by two vertical division at right angle to each other, resulting in 8 cell arranged in two tier of 4 cell each. The upper tier of 4 cell adjacent to the suspensor cell function as the initial of the root cap and the root epidermis, whereas the lower tier of the 4 cell facing the embryo function as the initial of root cortex. These cells further divides and develop to form the root cap, root epidermis and cortex of the radicle respectively. The division in the embryo, especially in the region where the cotyledon are to develop, results in the formation of heart-shaped embryo. The hypocotyle and the cotyledon by further transverse division elongate rapidly and enormously, so much so that the space within the ovule get limited for the expanding embryo. As a result, a marked curvature in the growth of the cotyledon is observed, which now curved and get placed within the ovule, usually parallel to the axis of the embryo. The mature embryo appear as a horseshoe-shaped structure in the longitudinal section, which is with a terminal stem tip bordered by two laterally placed cotyledons. In majority of dicotyledons, the cruciferous type of development is found. Some variation have been observed, such as the absence of suspensor in the member of Traphaceae and Orchidaceae, or suspensor become elongated as in case of Rubiaceae and Leguminosae. Thank you.